And here's our final experiment. That was a dialysis experiment. I hope you got a chance to look at there was one video that was a little longer and it had 3 different parts to it, and one part was the dialysis experiment. They had similar substances. Well, the way we run this dialysis experiment is that we fill a dialysis bag with a glucose solution, a protein solution, starch solution, and sodium chloride solution, and then we place it into distilled water in a beaker with water, and then we wait for an hour and watch which substance or substances would cross that semi-permeable membrane. And the way we will determine that is that we will take a sample of the beaker water and then test it for the presence of protein, glucose, starch, and salt. And for that, you need to know your indicator reaction. So I'm just gonna walk you through the results here. We use, in this particular experiment, for the detection of protein and glucose, we use these heuristics. They're very convenient because this is a dipstick, and you dip it into the beaker water, and it shows with a color change whether there's protein or glucose present. So the explanation of what a positive result looks like, it's a color change. In protein, it will be a color change, and it's yellow to kind of a green thing, green. And for the glucose, it's also a color change, but it's from like a turquoise, bluish, light blue, to brown. So it's more blue, light blue, to first green, brown, kind of color change. Benedict's test, that's one that we normally don't do in this lab number 3, in the diffusion lab, because we have the heuristics to test for glucose. That one also tests for glucose, and you will have a color change from light blue to red, orange, rusty red. It's kind of a precipitation. We'll get that in a different lab. Silver nitrate is used to detect salt, so specifically sodium chloride, and the positive result is a cloudy precipitation. White in color. Lugol's iodine is used to test for starch, and the positive result is dark blue. So a color change to dark blue. You would have been instructed to weigh your dialysis tubing before and after the experiment. And so usually before the experiment, since we add 10 milliliters of each of the solutions, protein, glucose, salt, and starch, it's about, plus the dialysis tubing, it's about 48 grams or so, let's say 48. Before, and then after it gains about 2 grams or so it would be about 50 grams, and so the net gain would be 2 grams plus it gains 2 grams. Okay, so now the experimental results. If you're testing your beaker water, the protein and the glucose, again we're going to do the heuristics. And the result of the dialysis for protein should be negative, therefore present in the beaker water, no. Glucose should be positive and present in the beaker water, therefore yes. The salt, the indicator is silver nitrate, so we have AgNO3, that's silver nitrate. The result after dialysis should be positive and present in the beaker water, yes, the salt. And the starch, the indicator is iodine, and result of the dialysis in the beaker water should be negative and present in the beaker water, no. Which substances pass through the membrane into the beaker water? So that would be glucose and starch, glucose and salt. based on your results, what have you learned about the size of the solute particles? Well, we have learned that glucose and salt are small enough to leave the dialysis bags through the semi-permeable dialysis membrane, versus protein and starch are too large. They cannot come out, and so therefore we didn't find them in the beaker water. So why do some leave or pass through the semi-permeable membrane of this dialysis tubing, and others don't? Well, it's clearly on molecular size. So glucose and salt are small. So glucose and salt are small by molecular size. Did water diffuse into or out of the membrane? It went into. What evidence did you use to make this decision? Well, it's the weight of the dialysis tubing. So it's heavier. It weighed 50 grams after versus 48 grams before. So therefore we gained 2 grams of water. Explain why water moved through the membrane. Well, because water moves down a concentration gradient that's called osmosis. And since we had a lot of dissolved particles in the dialysis tubing and distilled water or pure water on the outside, so now the water went from the area of high concentration from the beaker water into the dialysis tubing, and it's a small molecule, so it can pass the membrane, and so therefore the dialysis tubing was heavier afterwards because water went into the bag. And let me just draw a quick sketch of what this experiment looks like. So you have your dialysis bag right here, and it has the different components in here. So there's the salt, and then we had starch there, and we had glucose, and we had protein. And the only thing that we found after there, so then there's water up to here, and the only thing that came out was the salt and the glucose. So those you will be detecting afterwards in the beaker water. Okay, and this last chart, we don't need to do that. So I hope you got to see the videos. You can also try that dye experiment. I forgot to mention that, putting a drop of food coloring into hot water versus cold water. You can test that out yourself, or you can watch it on a video. It's kind of a cool experiment to do. It's kind of similar to taking a sugar cube and putting it into a cup with hot water versus cold water, and you see the difference. Definitely diffusion, the rate of diffusion is much higher when you have hot water as compared to cold water. Now you will have a quiz on this lab, so make sure that you know the indicator reactions, you know what the results were supposed to look like, make sure that you know it's solid on the terms, hyper, hypo, isotonicity. You should know what happens to red blood cells when you place them in 10% salt or distilled water, what these terms are called, chronation versus hemolysis, and make sure that you are firm on these indicator reactions that we talked about. So here, these indicator controls, what these things look like when they're positive or negative. And just kind of go over the questions here. You should be fine. Definitely start out with the important terms page on the very front there. So you should be fine. Let me know if you have any questions. But this quiz will probably be on Monday, so I'll post that as soon as possible. Oh, and the format will be just like the one, the first quiz, mostly multiple choice questions. You may have a matching set in there. Sometimes I put a fill in the blank or a very short, sort of a mini essay question, like a short answer question on the very bottom. But it's mostly multiple choice. And just make sure that you go over the lab. You should be fine.